It's really a tremendous pleasure to introduce Irv Epstein. Irv is internationally recognized for his foundational work in nonlinear chemical dynamics and pattern formation. His work has shaped how generations of scientists think about oscillations, instabilities, and emergent structure in chemical systems. His scholarship stands out not only for its depth and originality, but also for its clarity. He has a rare ability to make complex dynamics intelligible, and that has made him an extraordinary mentor to students and postdocs over many years. Beyond his scientific work, Irv has also been deeply committed to expanding opportunities for students, including through his role in founding the STEM Posse program, which brings students through college as mutually supporting cohorts, an approach that reflects his belief that talent flourishes best within structures of care, community, and shared commitment. Today's presentation is also a celebration of a different and important contribution. From the founding of the New England Complex Systems Institute in 1996 until this year, Irv has served continuously on the NEXI board, including many years as chair. That is an extraordinary span of commitment and help. Over nearly 3 decades, he helped guide NEXI through its development, its research directions, and its institutional challenges with steadiness, judgment, and generosity. Irv brought to NEXI not only his scientific insight, but also his deep experience as an academic leader at Brandeis University, including extended service as the provost. That perspective mattered enormously. He helped us think clearly about what it means to build a serious research organization, how to support students and postdocs, and how to integrate intellectual contributions with institutional responsibility. His guidance was especially important in shaping NEXI's collaborations with Brandeis over the years, including the supervision and mentoring of students and postdocs affiliated with Brandeis. So this talk here at Complexity Conversations marks a transition. Irv is stepping off the NEXI board, but he does so having left an indelible mark on the science, on the people, and on the organization itself. I am deeply grateful for his scholarship, his mentorship, his leadership, and his friendship. Please join me in welcoming Irv Epstein.
Well, thank you for the kind introduction. I don't think my mother could have done a better job. So this is a conversation, so I hope people will feel free, particularly if they don't speak chemistry, to chime in with questions. I'm particularly interested in patterns in chemical systems, though often as part of my collaboration with Yanir and Nexi, I've studied patterns in other kinds of systems, social, economic, biological as well. Today, I'd like to talk with you about two kinds of patterns, one in time and the other in space. My approach will be partly scientific, partly autobiographical, partly historical. The phenomena I'll talk about sometimes go by the name of nonlinear chemical dynamics, and I've actually, in collaboration with a former postdoc of mine, written a book with that title. Along the way, I'm going to focus on the interplay between deliberate design and serendipity, which has perhaps played an oversized role in this field. Most chemists focus either on making molecules or on figuring out how they're composed, synthesis on the one hand or analysis on the other. That is, chemists tend to be concerned about how to choose the right set of reactants and methods to get to a desired product, or about what that final product looks like. In fact, the past 5 Nobel Prizes in chemistry have been awarded for developments in either synthesis, most of them on this slide, or structural analysis. There is, however, another set of questions that one might ask. Uh, you might ask about what goes on along the way from reactants to products while the system's composition is changing, and about how fast this might be happening. After all, if your reaction takes a human lifetime to generate your product, it's not likely to be very useful unless maybe it's a biochemical reaction. These kinds of questions about the rates of chemical processes are the domain of something called chemical kinetics or dynamics. And although fewer chemists study these questions, and they have been the subject of fewer Nobel Prizes, they have been the subject of a few, including, interestingly, the first Nobel Prize in chemistry, which was awarded for the discovery of laws of chemical dynamics. But the Nobel Prize Committee quickly pivoted, and the second Nobel Prize and most of the succeeding ones was awarded for synthesis. Let me start with a video to show you the kinds of phenomena that I'm interested in. This is an oscillating chemical reaction, and hopefully, there we go.
Sequence. The reaction needs to be carried out with a magnetic stirrer. Take a glass with a first solution,
Irv, we can't see the video.
and
You
turn
have
on
to
mixing. Next,
exit.
the first solution is poured into the second solution, and finally into the first solution.
Can you see this?
Thereafter,
No,
the
you
reaction
have to exit the
first solution
presentation in
turns
order to
yellow,
see the
and
video.
then it becomes dark blue.
Hold
After
it.
that, the solution is
Okay.
You have to exit the presentation and share the video directly.
Okay. Let me move the video back a little. Um, can you see?
No, you have to exit there. Yeah, there. Now if you can see it exactly.
polarized, and the cycle repeats.
Can we see it now?
Yes.
Okay. So this is an oscillating chemical reaction where the concentrations change nearly periodically. So this is a pattern in time. It will go on for 10 or 20 minutes, but it will ultimately end up as in the dark blue state. All right, let me see if I can get out of this. Are we back to?
Yes, we're back to the presentation and we're back to the slide that you were on.
Okay. That will be the last departure from PowerPoint, so I think we'll be okay now. Okay, so this I think is visually compelling, but now a little bit of history slash sociology of science. For a long time, this kind of reaction was believed to be impossible, that most chemists and other scientists felt that it violated the second law of thermodynamics, which states in one form that the total entropy of an isolated system always increases over time. Now, you don't have to know what entropy is to understand that the idea is that if I have an isolated system like that beaker, that something is always going in the same direction. sometimes referred to as the arrow of time. Does anyone see why the reaction that I just showed might violate the second law? Uh, Yanir?
Yes, because it's going backwards and forwards, which seems to violate having a single direction of time.
Exactly. And until about a bit over 50 years ago, nearly all scientists saw it that way. These kinds of reactions are, at least in the lab, relatively rare, though if you think about it, you might argue that our bodies are full of them. Concentrations of many species change periodically due to chemical reactions, often on a 24-hour cycle. And so where's the contradiction here? I mean, the first people who discovered reactions like this, and there were two of these reactions, both discovered by accident, had trouble getting their results published because reviewers said this can't be. It violates the second law. So what the reviewers were missing was that, yes, if the reaction was doing what's shown on the slide here, uh, going from reactants to products to reactants to products, then the entropy would be going up and down. But in fact, that's not what's happening in the reaction you just saw and in the other oscillating reactions. What's actually happening is that the reactants and the products are colorless, and the species that are colored are intermediates, which are present in lower concentrations but are colored so you can see them. And so what's happening when A goes to C, the reaction's going rather quickly. Then when C goes to D, it slows down a bit. And then D goes back to C and it speeds up, slows down. And the color changes are associated with changes in the concentrations of these intermediates. But along the way, A is always going to B and carrying the entropy with it. So entropy is always increasing, sometimes faster, sometimes slower. And because the intermediates are colored, we get to see this, I think, rather attractive reaction. As I mentioned, if you wait long enough, the oscillations will stop because we run out of reactants. Same thing would happen in your body to the oscillating reactions if you stopped eating for a sufficient length of time. You would also come to equilibrium, which we call death. So the phenomena we're interested in are sometimes called far from equilibrium phenomena. Okay, now I want to take you back to 1973. Here comes a little bit of the autobiography. So 1973, I was a second year assistant professor at Brandeis who had been hired to do research in quantum chemistry, a fairly esoteric subject and totally unrelated to what I've just shown you. And at that time, there were two of these oscillating reactions known, both discovered accidentally. The first by Bray at Berkeley in the 1920s. And he managed to publish it in a fairly respectable journal, but the reaction was subsequently the subject of more papers aimed at debunking it than at investigating it further. The second reaction was discovered by Belousov in Moscow in 1958. And I'll tell you about that publication history in a minute. The reaction that I showed you is actually a more colorful hybrid of the two accidentally discovered reactions that was made by two high school teachers in 1973, Briggs and Rauscher. So Belousov's reaction ultimately had considerably more impact than Bray's, though Belousov didn't really get to enjoy it. He was never able to publish it in a journal. His only publication about it was in a one-page abstract in an unrefereed conference proceedings on radiation medicine. Uh, he did, however, write up his work and circulated it to chemists around Moscow in mimeographed form, a sort of samizdat. Uh, and one of these chemists in Moscow, Simon Schnoll, a professor of biochemistry at Moscow State University, had read this paper, and he had a graduate student named Anatol Zhabotinsky who was looking for a research project for his PhD. Zhabotinsky wanted to study periodic behavior in glucose metabolism in living systems, but Schnoll gave Zhabotinsky a copy of Belousov's write-up and asked him to see whether it might be worth pursuing. There he is. So Zhabotinsky went into the lab, made up the solutions according to the recipe, ran the reaction, and became really fascinated. And he decided that instead of studying glucose, he would do his dissertation on this oscillating reaction. He improved Belousov's recipe, he identified the source of the color changes, and proposed some ideas of his own about how the chemistry worked. He wrote up his results and sent them to Belousov for comment, and he received a supportive note in return. However, repeated attempts to establish personal contact with Belousov, even though they were both in Moscow, proved unsuccessful. Eventually, Zhabotinsky did manage to publish the work, and Belousov's contributions were lauded, but the two never met face to face, despite working only a few kilometers from each other. Zhabotinsky always suspected that Belousov, who had lost many friends in the Stalinist purges and who was employed at a secret military establishment, was wary of what might be considered extracurricular relationships. Belousov died in 1970 before his reaction really had its impact. Zhabotinsky left Russia in 1990, just after Perestroika, and he joined my lab at Brandeis and worked there until his death in 2008. Thanks to his efforts, the Belousov-Zhabotinsky, or BZ reaction, as it has become known, now serves as the prototype system for nonlinear chemical dynamics, and there have been literally tens of thousands of papers published about it. So Zhabotinsky did manage to get his work published first in some Russian journals in 1968, and later that year, he talked about it and showed a demonstration of it at a meeting on biochemical oscillators in Prague. And a professor from the University of Oregon, Bob Mazo, happened to be on sabbatical in Europe that year and attended the conference, saw Zhabotinsky's talk, got the recipe from him, and brought it back to Oregon, where he shared it with his colleagues, one of whom, Dick Noyes, a kineticist, became fascinated and decided to try to understand the reaction in more detail, because at that point it was just an accidentally discovered cute lecture demonstration. So back to 1973, I'm doing research in quantum mechanics. I'm teaching freshman chemistry at Brandeis, and my star student, Steve Jacobs, asks me if he can do a summer research project with me. Although he's very bright, I don't think he's ready to tackle a project in quantum mechanics. So I go to the library, when libraries still had journals and books, and I start browsing through the journals. I come across a recently published paper, which is shown here, on oscillating reactions, which I'd never heard of before then. And it turns out to be the key paper in the field. It's Dick Noyes who had gotten this recipe and worked in Oregon with two collaborators. And they did extensive experiments on the Bezier reaction and came up with a mechanistic explanation for the oscillations, how they occurred on a molecular level. So I read this paper and decide that this is a really cool phenomenon, and it seems like an ideal problem for an undergraduate to study. The only difficulty was that I'd been hired to do quantum mechanics, pure theory. I didn't have a lab. Fortunately, Ken Custon, a senior colleague, had a vacant bench in his lab and was happy to have Steve use it. So maybe first key bit of serendipity. Ken was also an inorganic chemist and provided valuable knowledge, which I didn't have, about the chemistry involved. So I asked my undergraduate, Steve, to go read the paper and then go into the lab and try to reproduce the experiments in the paper to get to know how the system works. After a few weeks, Steve comes in and says he's managed to reproduce everything in the paper with one exception. So the paper has all sorts of descriptions of the experiments and discussions of what's going on. And at the end, they have a section called additional observations. And one of these observations says that the oscillating
Irving's cell phone rang just as he.
The oscillating reaction can be completely inhibited by traces of chloride ion. We noted this effect, which had also been noted by Jabotinsky. And the key here is they noticed it when they tried to do measurements with a potassium chloride salt bridge, and no explanation has been developed. Steve comes in and says, well, he accidentally dropped some sodium chloride into the reaction vessel. And it was considerably more than you would get from the potassium chloride salt bridge, which just constantly leaks very tiny amounts of chloride into the solution. He said the oscillations did stop, as he would have expected, but then they came back, apparently unharmed. So I told him, do it again. See if this is real. So he came back, same result. So I said, okay, I'll come in the lab and I'll watch you when you do the experiment. Same result. So we spent the rest of the summer figuring out what was going on. And the key turned out to be the way in which the earlier experimenters had added the chloride. The salt bridge generated a very small amount of chloride, but it was being constantly replenished. It was dripping into the solution from the salt bridge. Steve had added a much larger amount, but only once. So as you waited, the reaction chewed up the chloride and then went back to happily oscillating. We worked out how the chloride reacted in the system, wrote it up, and got it published in the Journal of the American Chemical Society, the most prestigious journal in chemistry. This made me more interested in this sort of system. And the following summer, I had another bright undergraduate, Rob Kaner, and I asked him to look at the effects of iodine. For those of you who aren't chemists, iodine and chlorine are in the same column of the periodic table. They behave very similarly. And so my expectation was that iodine would do similar things to chlorine. Turned out that, as expected, iodine also inhibited oscillations, but only at high and low concentrations of iodine. At intermediate concentrations, it actually induced a different kind of oscillations. We again figured out what was going on, wrote it up, and again got it published in the Journal of the American Chemical Society. So at this point, I was beginning to find oscillating reactions more interesting than quantum mechanics. I mean, it's more fun when things turn color. And I began to think more deeply about the phenomenon. I found it disturbing that in spite of a lot of knowledge that was beginning to accumulate, there were only two of these reactions known, and both had been discovered accidentally. I decided that if you really understood the phenomenon, you should be able to design a chemical oscillator. Fortunately, I had just gotten tenure, so I was able to change my research direction without having to worry about job security. I came up with a scheme to build a chemical oscillator, inspired by a mathematical model developed in Bordeaux by two young chemical engineers, Patrick de Kepper and Jacques Boissonade. Uh, the model suggested that you run an autocatalytic reaction, a reaction that speeds up as the concentration of products increases. This is a relatively rare occurrence in chemistry, although it's the origin of the population explosion. In chemistry, it can lead to actual explosion because if the reaction starts going faster as you make more and more products, it can take off. And if you run one of these reactions in a flow reactor where you are continually flowing in fresh reactants and allowing products to flow out, and you combine that with a feedback reaction that prevents the autocatalytic reaction from getting out of control, the model suggested that you could get chemical oscillations. The model, however, contained no indication of how to find an actual chemical system that might behave this way. So I turned to my colleague Ken Custon, whose lab space I had been using, and who had also become fascinated by oscillating reactions, to plumb his knowledge of inorganic chemistry. And we were able to come up with some reactions that we thought might work. To implement these ideas, we needed some funding. And so I wrote a proposal to the National Science Foundation. It was rejected. I tried again, no luck. 3 times it was rejected, with about half the reviewers saying, great idea, this should be funded, and the other half saying, interesting idea, but it'll never work. On my 4th try, I got lucky and got enough good reviews to get a grant that would pay for chemicals, equipment to build a flow reactor, and a postdoc to carry out the experiments. I wrote to the leading labs in the field advertising the postdoctoral position and got 2 promising applications. One from Patrick de Kepper, who was one of the people who had proposed the model, and one from a young Hungarian, Miklos Orban, who had done some very nice experiments on the BZ reaction. I was torn about which one to hire when Patrick wrote to say that the French National Science Foundation, the CNRS, where he worked, had a program that would pay young researchers to spend a year in a lab abroad. So I was able to bring them both to Brandeis. They arrived in the summer of 1978, and by December, we had the flow reactor up and running and began to try out some of the reactions that Ken Custon had suggested. For a few weeks, we had some promising indications, but no oscillations. Then, late one Friday afternoon, serendipity reared its head again. Patrick came by my office and told me that before shutting down for the weekend, rather than pouring his solutions down the drain, he tossed the last of them into the feed for the flow reactor. And the reaction had begun oscillating. Unfortunately, he hadn't written down the amounts he had used because he was just sort of tossing them in there. And it took us another week to recreate the right conditions. But then we had it, the first deliberately designed chemical oscillator. We soon applied the approach, and more systematically and writing everything down, to several other reactions. And over the next few years, we and others using the method that we had developed, we're able to construct literally dozens of new chemical oscillators, where previously there had been only the two. So this is a sort of taxonomy of chemical oscillators according to what the key species in them are. And the only two that were known before we developed this method were this one, which is the Belousov-Zhabotinsky, and this one, which was the one that Bray had discovered in the 1920s. All the rest, and by now there are even more, were discovered once we had proved that you could actually design these things. Now, the behavior that we're looking at all occurs in stirred systems, so that the composition in the beaker or the reactor is the same everywhere in the system. The whole system oscillates together. In his earlier work, Zhabotinsky had shown that the BZ reaction, if performed in an unstirred petri dish, could also give rise to spatial patterns. And here's an example. So this is a petri dish containing the reactants for the BZ reaction. And it starts off and it's initially red. And then a little blue spot appears and then grows into a ring. And then you'll see how the system evolves in time. It forms these so-called target patterns, and they nucleate all over the reactor, and you get this very complex kind of pattern. Much of the interest in oscillating reactions arises from the analogy to biological systems. After all, as I mentioned, our bodies in some sense are just large flow reactors. We take in reactants, food, and we expel reacted products, wastes, while we generate periodic chemically driven behavior. The analogy carries over to spatial patterning as well. So this is the reaction that I just showed on the previous slide, the BZ reaction, after it's had a chance to develop for a while. This is on the right is a slime mold. It's an organism called Dictyostelium discoideum. And when it has sufficient nutrient supply, it exists as single amoeba-like cells that go about, do their business, reproduce, die, go through generations. But if you deprive it of its nutrient supply, it has a survival technique. It forms spores. And the spores can ride out long periods of deprivation, and then if you restore the nutrients, the spores open up and the cells resume their business. But during the process where the cells come together to form these aggregates, these spores, if you stain the medium, then what you observe are patterns like this, which I would argue bear a striking resemblance to the oscillating chemical reaction. And people have used insights from these kinds of reactions to understand the dynamics of cellular aggregation in Dictyostelium and other systems. So these patterns are spatiotemporal. Concentrations are changing in both time and space. Now I want to look at another kind of pattern in which the concentrations are constant in time, but they oscillate in space. Think about the spots on a leopard or the stripes on a tiger. These are periodic spatial patterns. How these patterns emerge and how animals develop in general is a fascinating problem. It even got the attention of Alan Turing, better known as the father of modern computer science and for his efforts in breaking the Axis codes in World War II. Turing actually wrote one paper on chemistry, and it was about how patterns develop in living organisms. He proposed, using several chemically unrealistic models, that a set of chemicals, which he called morphogens, that reacted together and diffused through cells, could account for the phenomenon of morphogenesis, that is, pattern formation in living systems. And this, I mean, perhaps even more than the existence of oscillations in time, is rather counterintuitive, since one expects diffusion to smooth out concentration differences rather than enhancing them. If I put a drop of food coloring in a beaker of water, the initial red drop over time just spreads out, and eventually I get a uniform pink solution. But what Turing was able to show with some nice mathematics was that if the system was also undergoing chemical reactions with the right kinetics and with the right relationships between the diffusion rates of the chemicals, the so-called morphogens, then pattern formation could occur, and you could get something where diffusion would actually enhance initial differences in concentrations and lead to a pattern. So Turing patterns occur in systems where the normal steady state, where the system isn't changing, is stable to perturbations that change across the whole system. So in a stirred system, this won't happen because if you perturb the system, the perturbations occur everywhere in the system. However, in a Turing system, the system is unstable to inhomogeneous perturbations, perturbations that are larger, that maybe increase concentration in one place and decrease it in another. And then the final structure you get is stationary in time, but periodic in space. So again, think spots or stripes. And it has an intrinsic wavelength. The distance between the spots or the stripes don't depend on the size of the system. And typically, the kinds of systems that behave this way have an activator. Think about the autocatalytic species in an oscillating reaction, something that tends to grow quickly over time, and an inhibitor, a species that tends to stop that growth. And in order for the system to show Turing patterns, the inhibitor has to diffuse much more quickly than the activator. So Turing's paper was published in 1952, 2 years before he committed suicide. It inspired some interest among mathematical biologists who incorporated some biologically plausible models and showed that Turing's approach did indeed lead to pattern formation. However, for nearly 4 decades, there was no experimental evidence for the existence of Turing patterns. They were a mathematical curiosity. So Turing publishes the paper in 1952. A bunch of mathematical biologists published some papers, most notably Gier and Meinhardt in Germany. But then 1990, in Bordeaux, Patrick de Kepper has returned from his postdoc at Brandeis, launched his independent career, and is now running his own lab. And he's studying one of the oscillating reactions that we developed at Brandeis, a reaction called that involves chlorite, iodide, and malonic acid, so-called SEMA reaction. And instead of running the reaction in a petri dish, he runs it in a gel, which they could feed fresh reactants into a sort of unstirred flow reactor. Uh, and um, and so they could maintain the system far from equilibrium because that would enable them to observe pattern formation over long periods of time. And so he has a graduate student, Vincent Caste, who is running these reactions. And one day, instead of seeing the target patterns or spirals like the patterns I showed you that they were expecting, um, Vincent saw a different kind of pattern that didn't change in time. And he showed it to Patrick, sort of like Steve Jacobs had come in with his observation and showed it to me earlier. And fortunately, Patrick had read Turing's paper and said, "Oh, this looks like a Turing pattern." He said it in French. And they went back and again repeated the experiments, did more careful measurements. And in fact, this was the first experimental validation of Turing's theory. And again, a considerable element of serendipity. So why had it taken nearly 40 years to find experimental evidence of Turing patterns? Well, in biological systems, there were no techniques that allowed one to follow the evolution of morphogen concentrations. You couldn't measure them. In chemical systems, one runs up against the requirements that comes out of Turing's analysis that in an activator inhibitor system, the activator needs to diffuse about 10 times faster than the inhibitor. And nature is cruel to us here. In aqueous solution, which is where most chemists do their work, all small molecules diffuse at nearly equal rates, within a factor of 2 or 3 of each other. So why was the Bordeaux group able to observe Turing patterns? It actually took us about a year to figure this out. The key was that in order to visualize the patterns, which depend on the concentrations of iodine and iodide in the gel, the Bordeaux group had mixed some polyvinyl alcohol into the polyacrylamide they used to prepare the gel. Polyvinyl alcohol forms a deep blue complex with iodine that makes the patterns much more visible than the pale brown color that occurs without it. And polyvinyl alcohol is a huge molecule that diffuses very slowly in water. So as it turns out, since iodine is the activator in this SEMA reaction, and it spends about 90% of its time attached to the polyvinyl alcohol, its effective diffusion rate is reduced by about a factor of 10, just enough to satisfy Turing's criterion for the ratio of activator to inhibitor diffusion rates. Once we figured this out, we were able to construct a mechanism for the system and do numerical calculations that showed that the SEMA reaction should indeed generate Turing patterns. This also led to our proposing an approach to designing new systems that could exhibit Turing patterns, again, going from serendipity to design, basically by running an activator inhibitor system in a gel reactor and adding an immobile substance that complexes the activator and slows its diffusion. Meanwhile, in the ensuing several decades, remarkable advances in biology have allowed scientists to follow the morphogens in several biological systems and demonstrate that the Turing mechanism is applicable there as well. The next slide shows the similarity
Irv,
between.
you're out of the slides. You somehow we're looking at you need to go to a different
All right, it's showing on my screen. Let's see, maybe the share is. I think someone else shared their screen.
oh, that's what happened.
Yep. Is it back yet?
There's no screen share now, so you can
Okay.
share again. Yeah.
We have it now.
Not yet. Does anyone, as far as yeah, now you have it.
Okay.
Yes.
Yeah. It was set on all participants could share, and somebody shared. Okay. So here is a set of Turing patterns. This is the SEMA reaction, the inorganic chemical reaction at two different sets of concentrations. This is a tropical fish, the chest fish, and I never remember which is which. I think this is the male and this is the female. So the way in which we transform from spots to stripes in the chemical reaction is we change the concentrations. Presumably, what differentiates the spotted male from the striped female in the fish is different concentrations of some presumably hormone type chemicals that are present at different concentrations in males and females. The Turing mechanism has now been applied to try to understand pattern formation, not just in chemical and biological systems, but to understand patterns of criminal activity in urban areas, galaxy formation, vegetation patches in desertification. It's become a fairly universally applied paradigm. All right, I'm getting toward the end. I think maybe just a few lessons and reflections. So one big lesson that has come out of this is that just as one can design and synthesize molecules, which has been sort of the traditional business of chemists, one can also design and synthesize dynamic phenomena. Design requires some kind of prediction, and prediction is not easy. As our sage Yogi Berra has stated, it's tough to make predictions, and retrodiction is significantly easier. Here's one insight into predicting a very complex system. But another very important notion is that chance favors the prepared mind, attributed to Louis Pasteur, that serendipity, well, unexpected things happen all the time. They become serendipity or favorable chance when you have some idea of what might happen. If Patrick de Kepper hadn't read Turing's paper, he wouldn't have known that those patterns were Turing patterns. When I started giving talks on oscillating chemical reactions around the country in those days when one could still carry chemicals and do lecture demonstrations before TSA came along, people would come up to me afterwards, and occasionally someone would say, Oh, I once saw something like that, but I knew it couldn't happen, so I changed the conditions and made it go away. Uh, So design is a form of prediction. In making predictions about complex nonlinear systems where there are likely to be variables that we can't follow or maybe don't even know about, we're going to be wrong some of the time, maybe most, but maybe not very wrong. And it's important to know, what being wrong will cost us. Usually, being wrong only maybe costs you some of your reputation, but it doesn't lead to death or explosion. And so we should be willing to try, and we should be poised to recognize these behaviors that we don't expect. And finally, don't underestimate the importance of the aesthetic. What lured me and I mean many of the others in this field, which didn't exist as a field 50 or 60 years ago, was that it's fun. These reactions are pretty. It's fun to look at these patterns. And I think that that has played a very important role, not only in this field, but in many other scientific fields. So let me stop there, and I'd be happy to answer questions.
Thanks, Irv. That was amazing and very good. This as a introduction to this whole domain. That's a very, very powerful presentation. Let's take some questions. Irv, do you want to stop your sharing so that we can see the people that are raising their hands? And Karen is raising her hand. So,
There we go.
it was
Yep.
for.
Stop share.
All right. Who would like to ask a question? I think that I'm sure that you had the experience that there was a period of time when it was still difficult to communicate about this. How long did it take before people were really willing to recognize this, or has it continued until now, I would say?
I think most people now believe. Um, Well, it depends where you start counting from. I mean, when I came along, I think there was already beginning to be receptivity. I think it had to do in part with um, global politics. Um, Initially, these kinds of systems were studied mostly in Eastern Europe, in part because they were cheap to study and they didn't have a lot of money for big equipment. And it was really when the BZ reaction was brought back from Moscow and Prague to the U.S. And started getting attention from American scientists. And a man named Ilya Prigogine did some very nice theoretical work on non-equilibrium chemical kinetics and got the Nobel Prize for it in the 70s. So around the 70s, things start really opening up.
Yeah, 70s was definitely a decade where new ideas were
Yep.
coming out. Renee, who would like to ask questions? You can.
Yeah, thank you. Thank you. Very interesting in the presentation, Professor Epstein. Do you know any connections with psychology, with the production of regular patterns in psychology? Because when you see, for example, the cycle of being awake and sleeping,
Mm.
when it gets disturbed, and
Yep.
I saw a connection when you were talking about the difference in the chemical reaction when you stir up the liquid and when you leave it stationary, the
Hmm.
different patterns that you see. I'm seeing studies about the effects of exercise, and I never thought about that the exercise just moves the chemicals in the body. So it probably it favors the oscillations in the body, and it probably helps to eat, having more regular
Yep.
sleep and regular eating, and whereas if you stay sedentary, there is no chemical
Mhm.
reactions or something, I don't know. Thank you.
Yeah. Well, yeah, I think all of these systems have some control parameter that you can tune and bring them from a stationary state into an oscillatory state, and then occasionally, in some systems, into a chaotic state. And if this control parameter is not within the appropriate range, then the system will just sit there in an equilibrium or pseudo-equilibrium state. And it could well be that exercise increases levels of some chemicals that act as a control parameter to bring the system into an appropriate oscillatory state. I mean, the other thing is that we and many other people have used these approaches to model the behavior of individual neurons as oscillators and looked at the interaction. One of the things I didn't talk about was what happens, excuse me, if you couple two oscillators together. This goes back to Huygens in the 17th century, who looked at the behavior of two pendulum clocks on a wall. But neurons are classic examples of coupled oscillators. And it turns out even if you couple just two oscillators, if they're nonlinear, they can generate a remarkable range of behaviors.
Yeah, thank you.
Thanks. Karen, go ahead.
Thank you very much for being here. That's very interesting. I was interested what you were just talking about, control parameters, oscillation, chaotic state, equilibrium state. You're in the world of chemistry and I'm in the world of education. And was a teacher in traditional education for many years. And then I went into a Montessori environment. And what I noticed is that I've been looking at Montessori through the complexity theory lens because the environment is so complex. And what happened is that I saw behaviors coming out of that complex environment that reminded me of Abraham Maslow's hierarchy of needs, where you see these amazing humane behaviors in kids coming. I had the middle school, so I saw 13 and 14-year-olds behaving differently. So my question is, do you know of places, people who are doing biological, evolutionary, human development kinds of things that would connect to what you're talking about regarding the parameter oscillation, chaotic state, equilibrium state? I saw that happen again and again and again in Montessori, and I'm writing a theoretical framework to try to figure out how to explain what it is.
I'm not aware. I mean, most of the people I hang out with are chemists and physicists who like to look at the simplest possible systems, and you've probably got the most complex possible system there. So, I mean, I think the overall framework fits. The issue is, what are your variables and what are your control parameters? And I mean, I think one could certainly make models that could mimic the behavior. The question I guess I would ask is whether they would also have explanatory or predictive power. Because I guess, yeah, the ultimate is you'd like to be able to design a system to exhibit the desired behaviors. And that, the more complex the system, the more challenging that becomes.
Thank you very much.
Any other questions that we have? We're a little bit past the hour. Marcus, I was hoping you would have one. Go for it.
Okay. Hi, Irv.
Good to see you.
Nice to see you. Yeah. So you started with these oscillating reactions, and then you moved into Turing patterns, which are stationary.
Yep.
So is there any deep connection between the two phenomena? Can you go from one to the other? For instance, can you make a busy reaction where these patterns just stop in the petri dish? And
The BZ actually is a source of considerable frustration because although it is the prototype system for most dynamical behaviors, we haven't been able to find a suitable complexing species that will slow down the inhibitor to give Turing patterns. So the Turing patterns have been found in other reactions, but there certainly is a correlation between oscillations and Turing pattern behavior because nearly all the systems I know of that show Turing patterns can also show oscillations in homogeneous media.
Okay. It's just a matter of adjusting the parameters.
Well, and being lucky enough that, yeah.
Okay, thank you.
I have one question, which is obviously you mentioned Jabotinsky and he came to the US and worked with you, but you didn't talk a lot about the early contributions that you made with him. Do you have something that you would like to point to?
Well, one which I'm sort of fond of, I'm not sure it's had enormous impact with, he was able to show the analog of Snell's law in one of these chemical systems, where if you had a gel or two pieces of gel that had somewhat different chemical composition,
and
you had one of these waves that I showed moving across the gel. When it went from one gel to the other, it changed the angle of propagation. It refracted just the way a beam of light refracts in going from one medium to another. So a chemical implementation of Snell's law.
Interesting. Very interesting. There's been so much discovery. The picture that is always in my mind about this is the phase diagram of all of the different kinds of pattern formation that one can achieve with the control parameters. And I always am thinking about the tremendous utility, but also the tremendous beauty. As you mentioned, it's amazing to see how things can be done when there's a rich dynamical system that can be controlled in various ways. I think we're at the end of our discussion today. I want to thank again Irv. It's been a long time. Irv, I was recommended to speak to Irv very early in my work in the 1990s, and Irv was tremendously helpful when I wrote my textbook, and he reviewed a chapter of my book and gave me corrections to it that were quite remarkable. One of the very few that really offered substantive and very informative improvements in the work. So I think I have a lot to be thankful for Irv for many years of collaboration. And I think it's clear that science has a lot to be thankful for his thoughtful contributions using both the kind of intellectual clarity, but also the receptiveness to what you encounter as you discover new things. Thank you, Irv. For me and for all of us, I really appreciate your joining us today.